what are your current position and field of interest? Could you please describe the group you lead, its facilities and technologies? What are the main objectives of the group, and what kind of research activities are currently ongoing? What are the perspectives for future research in the biomolecular field over the next years? And how might artificial intelligence impact research? What advances are expected, and how could they contribute to the development of new technologies and research approaches in this field? And now, Professor Riccardo Dalla Favera. Riccardo.
Okay, here I am, and I'm at Columbia University, like Raul, where I've been for the last 40 years. I have two jobs, in a sense. I direct an Institute of Cancer Genetics, which is a group of a dozen independent investigators working on different type of cancers or mechanisms that are common to most cancer type. And I direct my own group, which is focused now for decades on the pathogenesis of B-cell lymphoma. So my story starts from cloning human oncogenes, mapping them on chromosome, and seeing that they have involving chromosomal translocation, my Burkitt lymphoma. And for decades, the educated guess from cytogenetics or other approaches led to some example of mutated tumors, of which we and many others studied the normal function in B cells and the function when they are mutated. This brought to the attention very important biological players like MYC, BCL2, BCL6. This is the work of many labs, of course. But the question that remains in the air for a long time is how many mutant genes it takes to make a tumor or a lymphoma. And all this, as you know, changed with the advent of next generation sequencing. And now we know more or less the large cadre of mutant genes that are recurrently mutated, high frequency or at very low frequency, a long tail of low frequency mutant genes. So I think the effort of my group has been twofold, as it is today, to understand as much as possible the biology, the individual contribution of the most frequently mutated genes. This is with classic experiment in vitro, or modeling the mutation alone or in combination in conditional mouse directing the mutation to the target organ. That this case is the germinal center in the B-cell lineage. So this, as you know, in B-cell lymphoma, but many other tumors, has brought to a catalog of mutation that then are organized in classification with the attempt to have some prognostic relevance and also to identify targets for therapeutic therapies. This is an ongoing effort in many labs, because now we have thousands of genome exomes and full genomes available to study. I think in the case of my group, a turning point as what involves what Raul has mentioned, that, as you know, the coding genome is around 2-3% of the whole genome. And most of what we know in terms of biology of and genetics of tumors, B-cell lymphoma, but many tumors, is based on coding loci, copy number variation, missense mutation, truncating mutation, chromosomal translocations. But the rest of the genome is mute functionally in many tumors. And so I don't know if you saw our last work, but I think it's evidence emerging in many tumor types that there are functionally important, pathogenetically important mutation affecting non-coding regulatory domain. The mutation in B-cell lymphoma affecting superenhancer are one example, but there are probably many other type of regulatory regions that are involved. in my view, any recurrent there is tremendous noise, as you know, and Raul has mentioned, along the genome, the non-coding genome, especially high noise in B-cell lymphoma because of the activity of activation-induced deaminase. But there are clear in B-cell lymphoma as well in another tumor cluster of recurrent mutation that are really, really frequent. Our attention is on those because in my career, whenever a tumor keeps on hitting, there is something important. And we can action this recurrent mutation functionally, looking whether they are a regulatory element, whether they affect the chromatin domain, but there are many, many different approaches. I think this looking at recurrent mutations in general in cancer in regulatory domains will be very important because it will complete the classification of tumors, therefore complete or progress the understanding of the biology, which in many cases is still obscure just based on coding mutation, will allow to classify that better prognostically, will identify new therapeutic target. I think my last point is I expect, I'd say from this is our interest also, to apply functional analysis of regulatory domains, not only to somatic mutation, but we want to reassess, and I think many other people may have the same idea, GWAS data. There are a wealth of GWAS data on mutations, single point mutation or others that have a strong association with various type of malignancies. And there are already examples in type of cancer, but also in other pathologies in which you functionally analyze those mutations that have remained mute for decades, that in fact they do sometimes involve regulatory domains. So this is on a strong genetic emphasis that doesn't exclude other components, but I think as Bernard has said, I think the genetic of a tumor largely instructs the microenvironment and the microenvironment response. So I think there will be another step in the understanding of genetics and the whole biology. So of course, artificial intelligence, all the new technology, single cell analysis, and a lot of bioinformatics capable of reducing the enormous space of mutations into a reduced space of networks and pathways. I mean, that's, of course, an explosive field that will help a lot, but this has already been emphasized. So that's more or less what I have to say.
Thank you very much, Riccardo. You used the term, which is quite familiar to me, classification.
Yeah.
You know, in 2022, we had a great debate concerning classification of hematolymphoid tumors. I think that the future is more in the sense of a genetic classification, more than a conventional classification, which was the matter of content between the ICC and the WHO. What do you think about?
Well, I think, this is work in progress. I don't think the genetic by itself will ever be the only thing, although
no, sure.
Also, but again, then we have to know more about genetics because, for instance, in our field, if you take the fantastic work by Ship and Stout on genetic classification, a big fraction of tumors cannot be classified, and even the one classified are classified on association that sometimes are not as compelling as you would. So I think there's a lot more there to be digged.
Thank you.
But this doesn't exclude that a major component of a classification may be determined by description of the tumor microenvironment, and that the tumor microenvironment itself may become a target for therapy as opposed to directly cell autonomous to the tumor attacked.